Stem cells, these special cells that can be magically converted into other cell types of the body. So imagine you have a patient who has lost some of his heart cells due to heart attack. The heart cannot function anymore. But you can use these stem cells, make heart cells out of them, and replace the unfunctional cells with these new ones. You can save patients' life. So wow, there are so many diseases we can cure with these stem cells. But wait a minute, how far are we from reaching that goal? Safety is a big concern when we want to use these stem cells for any human body applications. Today, I'm going to talk about a specific safety concern. The first step for any cell therapy process is to grow your cells to have billions of them per patient. But research has shown that in around 34% of cases, when you are growing these cells, there are some cheater cells emerging in your population. These cheaters have some genetic mutations, enabling them to kill your normal cells and overtake the whole population. But what is wrong with these cheaters? They have lower efficiency when you want to convert them into other cell types, and also they have some similarities with cancer cells. So the standard protocol is to discard the whole batch of your cells if you see such cheaters emerging. But what about the whole time, energy, and money that will be wasted? Just think about that patient who is in immediate need of receiving these cell products. Here is where my project comes in. In my project, I try to help with the issue of having these cheaters in our cell population by answering some remaining questions in the field, such as, what are the types of these cheaters that emerge in our cell growing systems? How do they interact with normal cells? And how we can control their emergence, or at least growth, by controlling system parameters. One of these parameters is agitation speed. In these systems, you need to agitate the cells to have higher quality products. And I believe the speed of this agitation has a key role in emerging these cheater cells. Another factor is when you want to grow these cells, you start with single cells or cell clusters. And if clusters, what size? The goal of my project is to find optimum ranges for these parameters to avoid having these cheaters in our cell population. Hopefully, by finding these optimum ranges, we can make these life-changing products safer. Thank you.